Thanks for having me and thanks for sticking it out until the afternoon. I know it's hot in here, but today I'll be talking to you about moving drug pathway models towards mechanism. And just since it is sort of like a training, is it on? Oh, okay. A training retreat, I just wanted to follow up with part of the introduction in that I did have a somewhat non-traditional postdoc training period where I started at Stanford as a traditional postdoc and spent a couple months at the FDA, and then did an entire year at Genentech in a company and then went back to Stanford. So you'll notice that some of the questions that I'm asking and the problems that I'm addressing are influenced by that career path. And you're welcome, I welcome you to ask me questions about the training and those environments in addition to the research as well. So when I started my postdoc, many of you will be familiar with this, but one of the problems that I was aware of and somewhat interested in is this idea that therapeutic development success rates are often less than 10%. So what you see on the bar chart is the different phases of the development as you move from left to right. And then on the far right, what you see is the kind of overall likelihood of success. And so it's a lot of work. It goes huge R&D pipelines, and then there's more time and energy spent in the actual development pipeline. And at the very end, what you see on the far right there is that for across all different disease indications, the overall total was about 8.4%. So less than 10% of drugs that start in the clinic make it to the market to patients. But what's interesting is that on the far right there, the 25.9%, that blue bar, it's the idea that if you have a biomarker for your drug, those tend to have a higher likelihood of success. And the high level takeaway there is just that if you have some mechanistic evidence that associates what your drug is doing to the underlying disease pathology, the takeaway is that you can be more successful. So then just looking at a very high level reason for why many of these drugs fail, it's often for intolerable safety or insufficient efficacy. So this is another bar chart, one of many that have been plotted in this space. And if you just look at the blue and red bars, the kind of far left, the left two bars there are efficacy and safety, and that those kind of account for a large proportion of drug failures. And then just to give you a little bit more information, the phase 3 is the blue bars, and that's sort of the clinical trial progression of phase 1, 2, and 3 development. So things will sometimes fail early in development. And then the red bar is the NDA, which is a new drug application, or BLA, which is a biological licensing association. So those look at small molecule drugs as well as protein or biologics. And essentially just, you see this similar-ish trends in both styles of therapeutics, and that again, safety and efficacy and the gross category level are kind of behind this. And then just to give a little bit more motivation, drug adverse reactions are among the top 10 reasons for death in the US, and they are estimated to cost somewhere on the order of 10 billion annually. So as an engineer, one of the things that I thought about when I looked at this particular challenge is that I think one of the limitations in the space and what's currently in the industry and how drugs that are successfully going to the market is that many of them are very considered first order. And so you may have heard of the one drug, one target, one disease paradigm. And the way that I think about that in like a very cheesy engineering cartoon here is that you have a drug, which is a triangle, and I'll have many orange triangles in my talk. That orange triangle binds one protein, that's a black circle in this drawing. And then that black circle, that protein or gene is associated to one disease. So you often see something that's mutated or overexpressed, and the idea is, okay, in a single-minded focus, we're gonna try and change that one particular disease gene or disease protein. And so in an oversimplification of the process, there's a lot of R&D that goes into looking at different types of evidence, different literature sources. And at the end of the day, what you get is sort of a ranked list of candidate genes, if you will. And you don't even have to read them, but the red arrows at the top of the list. And then usually it's some aggregate process of that evidence. And then you take that lead candidate, and that's what goes into your development pipeline. But there are several challenges with this approach. One is that many diseases are polygenic. So there isn't often one driver mutation that is the sort of golden ticket for improving patient outcomes. And then further, we also know that many drugs bind off-target proteins in the small molecule space, or even further, that when you perturb a gene or protein's function, it often has ripple effects through protein networks. And then one of the other things I just thought was sort of intriguing is that if you think of the cell in these like cartoon images, I'm not even quite sure how you engineer specificity, given that this is often what your molecule or biologic interfaces with. So the cell is a really crowded space. There's a lot of things to bump into before you even have your desired effect. So the other perspective that I brought to this process is thinking about how drugs work and how they're designed is that I think of the cell as sort of like a pinball machine, if you will. And so if anyone hasn't played pinball, there's a picture of that in the middle. And it's an arcade game where you have a ball that's introduced to a set of bumpers, and you kind of have to bounce off those different bumpers to score points. And that's kind of how the game works. And so at a very high level, you can think about the cell in a similar way, where when you introduce a therapeutic to the cell, it uses the protein interaction network to cause efficacy or have some favorable disease modifying effect. But the same protein interaction network is used to influence intolerable safety or unintended side effects. So for today, I'll tell you 2 stories just to keep it somewhat short so there's time for questions. But the first part, I'll talk about using protein-protein interaction networks to describe drug effects, and I'll talk about the PathFX algorithm that I worked on as a postdoc. And then I'll talk about leveraging real-world evidence to test drug network proteins, and specifically with a case study in detecting rare drug-drug interactions. So the algorithm that I developed is called PathFX, and it's a networked tool for discovering drug phenotypes. So I have the similar cartoon on the far left where you have your drug, you have your cell pinball, and then you have your sort of phenotypes at the end. And the way to kind of like decode this particular process is at the very top, PathFX starts with a drug, so that's my orange triangle. And then you have those red circles, and those are the drug binding proteins. And then in the middle, what it does is it adds gray circles to the network. And what those gray circles represent are downstream proteins that are likely to be relevant to the target's function. And I don't have all the math in the main slide, but I can talk about it afterwards. But at a very high level, the way that PathFX makes decisions as to which proteins to add is using the amount and quality of evidence to support the interaction. So you can integrate data from multiple databases and multiple data sources. And for proteins that have multiple citations, multiple screens, maybe detailed biochemical assays, those proteins have an edge weight that's higher than those that may have just dropped out of a yeast-2 hybrid screen and don't have further validation. So the way that you use this is you say, how much data do I have to support those interactions and which are the most likely relative to my network as a whole? And in the third step, you take the union of all of those sort of subnetworks. And then in the last step, I add a bunch of green boxes to the network. And that's basically an enrichment exercise where you say, if I've selected these 25 proteins out of my tens of thousands of proteins in the entire interaction network, what are the statistical possibilities of getting an association to diabetes genes or cancer genes? And that's just a modified Fisher's exact test where you start to do this phenotype enrichment. So to give you a more concrete example, I always like to start with metformin because metformin is my favorite prototype drug. And so again, you see an orange triangle at the top that's meant to represent metformin, the drug. There's red circles below that, which are the drug binding proteins that are in my interaction network. And then there's a whole bunch of gray circles of additional downstream proteins and gene variants, which PathFX adds, given that, again, doing that same exercise of how much data supports these interactions and how likely is it that these protein interactions are relevant to my drug targets. Phenotypes that are associated with this network more than you'd expect by chance compared to the entire interaction network. And to make a lovely story and a lovely photo, what's in the blue box is that I've identified 2 diabetes-like phenotypes that are associated with the network. And as reference, metformin is an anti-diabetic. And so for me, it says, okay, great. There's a way to sort of connect the dots from metformin's drug targets to diabetes-associated genes. So it's intent to treat disease. And so in the original paper, what I did was also do this analysis for an additional 238 drugs and a total of 1,756 drug disease pairs, because some drugs are on the market for multiple diseases or are used off-label for multiple diseases. And for about 60% of the 238 drugs or about 30% of the drug disease pairs, you could do this in a data-driven fashion by keeping PathFX blind to the intent to treat disease. You just say what phenotypes are associated given the amount and quality of evidence, and how does that align with what the drug is doing in the clinic. So another important takeaway from this particular exercise was it was just affirmation that a network paradigm could find meaningful associations for some drug context, if you will. And so the cartoon on the right is the paradigm I introduced before, where it's sort of one drug, one target, one disease. And the PathFX paradigm is summarized in a little cartoon on the left, where it's one drug, many proteins, many phenotypes. And by doing that modeling, you can still start to describe drug effects. And so it was just an initial question to say, can we use networks to describe these effects? Is there enough information for us to do sort of further analyses? Okay, so then one of many questions that came out of this was, does the network represent the drug's mechanism? Because it's nice to be able to associate metformin with particular diabetes-associated genes, but does that mean something for how the drug is functioning? So I'll go ahead and pivot to the second part of the story, where now to try and get at understanding what, if any, of those downstream proteins are relevant, I started using real-world evidence. And on a particular case study I focused on is in detecting rare drug-drug interactions. So one of the things that I thought was interesting was when I was both at the FDA and in industry, one of the ways that they regulate drug-drug interactions. So basically what that means is that if you have a patient who takes more than one drug, sometimes the combination of them causes some non-intuitive effect. And you can also imagine that clinically, when you're doing a clinical trial, it's actually not feasible to test every combination of drugs in a patient. And they do the best that they can to assess it through various experiments and animal models. But again, it's not feasible to test everything. And there are sort of rare combinations that you just can't anticipate during that development process. But the way that they primarily regulate those drug-drug interactions is what I describe as a shared protein mechanism. So the FDA has guidance on industry, and they say, okay, if you're developing drug 1, we already know that drug 2 binds that protein. We need you to test drug binding of your drug to this known protein. And those known proteins can sometimes be targets, transporters, or enzymes. And so many of the pre-tested or pre-anticipated drug-drug interactions are when they say, well, we have this known metabolizing enzyme. We want to know if your drug binds this enzyme or this transporter, and if it does, that'll affect these other drugs that are processed by that enzyme or transporter. But again, the idea is still that they look for sort of shared or competitive binding. So again, being a network person, I said, well, that's interesting because if I think that drugs actually work through these network mechanisms, it's actually possible that they interact via their protein-protein interactions. And so the idea that I tried to test here, which served two goals, was can you sort of predict drug combinations when they have a shared downstream protein? And that gets at sort of like, are there other mechanisms for drug-drug interactions? And then also allows me to test, if there's a protein downstream of metformin, does that seem to have any influence on what metformin does in patients? Okay, so one of the first things I did was reclassify drugs by their downstream proteins. So in the cartoon on the left, so for this particular scenario or the cartoon, I modeled 5 drugs. In this case, they're all associated with sepsis, so that's what is in the yellow box. And then I separated them left and right and tried to highlight for the drug networks on the left, there's a protein A that's downstream of drugs 1, 2, and 3, but protein A is not in the networks for drugs 4 and 5. So I'd call that on the left would be the A network class, and then the right would be the non-A network class. And then you go back to the original, so I use Drug Bank, which has a repository of drug to protein binding information. And then you predict drug-drug interactions, but with one caveat. So the drawing shows, I think, just A1 and A2 there, and they both are known to bind protein A, but A1 is eliminated because it has a shared target with drugs 1 and 2. So the really important thing here is that when I derived these drug-drug pairs, I didn't have any documented evidence that drugs A2 and 1, 2, and 3 shared any protein binding. The other thing that's important here is that drug A2 is not already associated with sepsis. So again, trying to find a non-phenotype associated drug that might affect a phenotype. And then to take a step back, to go to the data that I actually used, it was data extracted from drug labels. And so I have a little cartoon here where we have a whole bunch of triangles on the left, and that's just a sampling of those drugs. And just to give you a sense of that scale, there were 1,970 drugs in the total data set. And then there were 34 adverse reactions or severe side effects. And I did this in collaboration with one of my colleagues when I was at the FDA. And we picked these particular severe adverse reactions because they're of highest concern for FDA regulatory review. And then you get a sense of which ones are there, but to say that some side effect and drug interaction studies, you can have things like headache or rash, but we kind of gravitated towards the more serious side effects because one, they're pertinent to regulatory review, but it also narrowed the scope somewhat. So that gives you a sense of the starting data that I used for network modeling and then later for predicting the interactions. So the actual adverse reactions come from the drug labels. So on the labels, there's warnings, box warnings, and precautions. And another reason for using that is because in regulatory review, you can kind of assume that the FDA is like equally as concerned about like myocardial infarction for any and all drugs. And so they are kind of tested to this like a similar level of scrutiny. And so one of the challenges in network science as a whole is that you often don't have what I call as drug negative effects. So I showed you that lovely network with metformin, and there's a whole bunch of other phenotypes in that network, but metformin is not going to go through another randomized clinical trial to test for ovarian diseases or IBD or neoblastoma, whatever you've predicted, right? But the interesting thing with severe side effects is that if they're on the drug label, there was probably sufficient evidence to associate that drug with a side effect. But then you can also say that if the side effect was not on the drug's label, that the FDA cares a lot about sepsis, pancreatitis, myocardial infarction, that it's likely not causative for the side effect or exceedingly rare. So that was like an opportunity to have negative drug cases. Okay, so I think I already talked through these bullet points, but I just want to remind myself that we needed to have drug negative effects, and then this idea that drugs without a labeled warning are likely not causative. So then to give you a sense of, the punchline's there, so I'll put the figure up. But I wanted to compare the analysis to TwoSides, which is another database of drug-drug interactions. And this was done electronically looking at case reports where when a patient takes a therapy, if they have a side effect, usually industry, sometimes pharmacists will report into the FDA spontaneous adverse event reporting system. And it's a really interesting resource because you can imagine that either from a pharmacist or a drug perspective, the incentives are to go ahead and report. You don't have to do a lot of validation. There's not often a lot of follow-up, but the FDA tracks these reports and they say, okay, well, actually, you know what? This drug has been on market and we're getting a lot of reports for this side effect. And that's how they sometimes will know to intervene or to react. But it's also a noisy data set because you might say, okay, well, this patient's having like a hypoglycemic event, but you know what? They might be diabetic on a new medication and just like calibrating what's happening. So there's, again, many confounding factors in those types of data. But what TwoSides did is they used what's called, like at a high level, sort of propensity matching, where they said, okay, well, these patients all seem to be on metformin and like paroxetine or something. So you kind of can stack and compare similar-ish patients so you can recalibrate those scores. And then you say, oh, maybe the hypoglycemic event is not like excessively happening. It might just be associated with some of these patients who have these other comorbid conditions. And so the nice thing about TwoSides is it's regarded as one of the best example data sets of drug-drug interactions. And so it'll say drug A, drug B, and then the potential side effect. And so the first thing to do here was just I wanted to check of my network predictions, do any of them show up in TwoSides? And the number is a little bit small, but just to kind of go through the scale of the analysis at the top, again, we started with 1,970 drugs across 34 adverse reactions. Of those 1,900 or so, 1,100 of them were in drug bank, and about 970 of them were connected in my interaction network. So I had to filter out some of those to do this prediction. I had about 424 drug networks connected to 24 of the adverse events. So for some of the adverse events that the FDA care about, one of them, for instance, is suicidal ideation. They don't really have a good pathway annotation for that particular adverse event, so I'm not able to draw a network for it. But then we end up getting 172 network classes across 12 adverse reactions. So again, I was trying to look for multiple drugs that shared connections to the same adverse event. And then using that with the same sort of drawing with other drugs that are not associated to the side effect, you can actually make about 19,000 drug-drug interaction predictions. So then if you filter that where, again, because I said I wanted to eliminate that shared protein mechanism, there's about 6,000 drug-drug interactions, which include about 5,200 drug-drug pairs, because some drug combinations map to multiple adverse reactions. And then if you filter those using what's in case drug-drug interactions, which are in TwoSides, there's two things that I wanted to point out here. So for that 5,200 drug-drug pairs, there's a lot of them that are just not prescribed. Like there's no medical scenario where you would give drug A and drug B. So I first had to filter those and say, well, are any of these prescribed in the real world? Because I could predict a bunch of drugs that would just never, ever meet a human in the clinic. And so that left about 405 combinations with 457 disease drug-drug interactions. So the data set got smaller, but of those 405, 368 of them had a case report in this data set of TwoSides. So when we eliminated those that had a shared protein binding mechanism, using a downstream protein was pretty successful at predicting those additional rare drug-drug interactions. So then further filtered that because the goal then is to say, okay, well, the prediction is nice, but is there any evidence to support that? And so I had to narrow it down to about 60 network class predictions to use in the electronic health record. So then the last piece of data that I'll talk to you about here is actually going back to the health record to say, do we see this in patients? And just to remind you, these are the network classes and then the prediction scheme. And I just wanted to put that back up there because I had to make a couple of comparisons to measure if any of these interactions were real. And the first is to just compare the baseline, because maybe drugs 1, 2, and 3 are more risky than drugs 4 and 5. And so the first measurement I made in the health record is for patients who are on class A or non-A class drugs, what's the differential outcome in sepsis? And then the second measurement I made was drug A2 following a prescription or exposure to drugs 1, 2, and 3 compared to drug A2 following an exposure of drugs 4 or 5. And the goal there is to say, okay, well, if the combo, you want to first understand, well, is it just the fact that they're all taking a second drug because there are more side effects associated with more drugs or taking more medications? But in theory, if the network prediction is correct, it should only alter the risk for sepsis relative to that baseline class. So this is just simulated data, but what I'm going to show you are two hazard ratio plots. And what you see is that you'll measure the baseline hazard ratio and then the other hazard ratio. And for lack of a better term, I'm calling it a hazard ratio just to say what is sort of the altered risk for this outcome relative to the baseline. So I'll give you two case studies. So the TEN network class versus non-network class was measured for pancreatitis. And what you get here is the no combo effect, where there's about a hazard ratio of 0.5. So it seems like there's a slightly lower risk for the TEN network. And in this case, TEN could be that you had T53 EDNRA or NF-kappa B IA in your downstream network space. But then if you add aspirin to those drugs, the risk relative to the other class increases, in this case for pancreatitis. And then the other example is ADRB2, which was a downstream protein for many drugs associated with sepsis. And then you see without the combo, relative to the combo, there's again a slight shift in the risk for sepsis. So that's kind of the end of that story. I have a few other validations that aren't in this slide, but the takeaway for me was just the ability to sort of test downstream proteins using electronic health record and sort of getting creative with designing those experiments. It both had sort of implications for understanding rare drug-drug interactions, but again, going back to say, are the downstream proteins useful in identifying what the drug is doing? So I'll highlight just a few questions, but of many, but some of the other things that we'll be looking at or following up with this particular study is understanding drug combination effects on disease, because the formalism still holds. You can still make predictions, not just for side effects, but also for disease modifying effects. But part of that process is actually trying to understand drug efficacy negative effects. So I gave that example of, these drugs all cause sepsis, but these drugs that don't have it on their label are likely not causative for sepsis. But we'll have to use somewhat creative approaches to get at, well, if I predicted that metformin works in cancer and IBD and depression, how do I actually measure whether those are real or whether those are sort of null effects? So I'm going to go ahead and end here. I just want to thank people at Stanford and at the FDA who helped with this particular project, and thank you for your time. And I'd be happy to take any of your questions.